This is Chapter 10, Muscular Tissue. This will be new material for exam number 4. This will not be included in exam number 3. This will complete with chapters 11 for the muscular system, and then finish off with nervous tissue in Chapter 12. As we go through the online version of my PowerPoint presentations, there will be questions interspersed very similarly when we had met in lecture. We would break up in small groups to answer questions. I'm going to ask you to do instead is to answer these questions at the end of the chapter. You'll see them interspersed, and I'll give you a brief pause in between before I go into more detailed examination and answers on those particular questions. So again, I'll have you fill those out on a Word document and submit them as part of your assignment. Starting with muscular tissue, we're going to talk about the basic structure and function. So a lot of this will not be new to you. It'll be something we'll kind of went back over on Chapter 3 and 4 back in the earliest part of the semester. We'll talk about the 3 different types of muscular tissue, which include skeletal, cardiac, and smooth. We'll examine a little more closely into the neuromuscular junction, and we'll go a little more in depth with that once we get into nervous tissue. We'll talk about different types of energy used in muscle cells, and also how the nervous system in particular controls muscle tension and why tension is so important. Of course, 3 types of muscle tissue: skeletal muscle, cardiac muscle, and smooth muscle. And I'll let you peruse through this on your own. This is basically a static slides for where you normally find skeletal, cardiac, and smooth muscle. Of course, cardiac find only in the heart. Smooth muscle you find mainly lining any kind of orifice or any kind of canal, such as the GI tract. Skeletal, of course, is all voluntary, which include all the major muscles that you use for motion and for posture, and of course for voice control. So look at the different characteristics of the muscular tissue. Of course, skeletal muscle is found all along the skeleton, and the majority of your muscles are going to attach to bones, with a couple of exceptions, especially the muscles around the face and any sphincters. They don't necessarily cross or attach to bone, they attach to other soft tissues. Whereas cardiac muscle tissue is only found in the heart, and of course visceral or smooth muscle is found around the different hollow organs, such as the GI tract, the respiratory tract, and also the urinary tracts as well. Skeletal function is to move bones, or actually to hold bones in place, in particular for posture. Pump blood is the function for cardiac muscle, which would be no surprise to any of you. Visceral muscle, smooth muscle, has a lot of different functions. In particular, it does what's called peristalsis, where it allows movement of food one way down the esophagus, and also push a bolus or any other kind of foodstuffs back through the smooth muscle, through the stomach or through the small and large intestines, and of course out the anus. The actual appearance: skeletal muscle will be multinucleus, so more than one nucleus per muscle cell or muscle fiber. They're also striated because of the overlapping actin and myosin filaments. Cardiac muscle has one nucleus. They are striated, but also have the very unique characteristic of having intercalated discs, which allows electrical transmission of an impulse from one cardiac muscle cell to the other. It's how we keep a rhythmic beat with the heart. And of course, the visceral smooth muscle is only one nucleus with no striations. We'll actually look at the different components of what contractile tissue we have in smooth muscle. Is not aligned the same way as you would see in skeletal or cardiac. Of course, skeletal is under voluntary control. Up to a certain point, there is some involuntary control here and there just for keeping up a muscle tone. Of course, you know involuntary control for cardiac and for smooth muscle. We have no control over this. You can influence the cardiac muscle tissue up to a point, but you simply cannot tell your cardiac muscle to stop contracting. And let's look a little more closely into skeletal muscle tissue. So as you can see here, basically your tendon that attaches the bone to the actual belly of the muscle is continuous to the periosteum. It's a very strong connection, actually stronger than the bone itself. We'll talk about different types of injuries called avulsion fractures, where a lot of times the tendon attaching to the periosteum will not tear, the bone will actually break first. Not saying you can't tear the tendon more or less, but a lot of times you'll pull part of the bone off with it if there's any kind of injuries. We'll talk about a certain type of injury in just a couple slides. So basically we have a breakdown of the tendon as it goes to the belly of the skeletal muscle. It's surrounded by what is called the epimysium. And then within the epimysium you have certain little fascicles, which when you get to the hamstake lab, you'll actually be able to look at individual fascicles. We have a donated video from Dr. Mia Tamont, who's also an instructor here at Rowan University. She was nice enough to lend us her dissection videos of the chicken wing as well as the ham steak. But she'll show you how she's tearing apart individual fascicles, and they're relatively small. Each fascicle is going to be surrounded by what's called a perimysium, and then contained within perimysium are going to be multiple muscle fibers. These are the actual cells that make up the muscle. The muscle cells are in turn going to be surrounded by what's called an endomysium. So 3 layers of protective coverings will be the epimysium, which covers the entire muscle, which is continuous with the tendon and the periosteum. You have perimysiums that will surround each individual fascicle. You have an endomysium that's going to surround the actual muscle fiber or the muscle cell. And if then found within the muscle cell, you'll see the striations that will be comprised of the actin and myosin overlapping filaments. You will see within the muscle fibers individual nuclei, as well as special structures such as the sarcoplasm, sarcolemma, and also the transverse tubules. And then within each myofibril will make up the individual filaments. So you'll have contractile proteins, you'll have regulatory proteins, and you'll also have structural proteins that help knit everything together. So how are muscles formed? So just as you saw back before with bone cells as well as cartilage cells, they start out as blasts. So these are myoblasts, so they're akin to osteoblasts and chondroblasts. Once they start to fuse, they start to form into skeletal muscle fibers. Satellite cells will help nourish those particular muscle fibers as they continue to grow. One particular point though with myoblasts, once they start to fuse, then all of a sudden the muscle fibers lose their ability to mitotically divide once they become mature, and we'll talk about why that's so important later on in the PowerPoint. So look at some specific structures of the actual muscle cell. So as I mentioned before, there's the perimysium around the fascicle. You have satellite cells that help nourish the individual muscle fibers. Of course, surrounding each muscle fiber, you have the endomysium. You see a couple of nuclei that kind of get pushed out to the side as the majority of the cytoplasm, or the sarcoplasm in this case of the muscle cell, is going to be packed with contractile proteins. You also see a lot of different structures such as mitochondria. You'll see the sarcoplasmic reticulum, which is a differentiated version of the smooth endoplasmic reticulum you find in a typical animal cell. These are going to store basically in terminal cisterns. You're going to find a lot of calcium stores that allow the muscles to contract. You'll see different mitochondria, and very important for cellular respiration as far as producing enough ATP, which you know is integral to causing any kind of muscle contraction. And if you look further down into that diagram on the lower right-hand side, you'll see the sarcomere. This is the basic unit of a muscle cell. That's what allows proper contraction, which we'll go into a little more in depth in a few minutes. But basically, you'll see the thick and thin filaments, the myosin actin filaments, respectively, that basically allow the sliding filament mechanism, which we'll talk about in just a few minutes. This is a very simplistic view of what all the typical structures you would find in a muscle cell. You have the sarcolemma encompassing the entire cell. You have the sarcoplasm containing all of the different structures therein. You have the sarcoplasmic reticulum, which is your primary calcium store. If you notice, between two of the sarcoplasmic reticulum, you'll find a transverse tubule, very important for when we see any kind of action potential being generated across the sarcolemma, usually once we get some kind of nerve impulse from a axon. Each individual muscle fiber is going to have a lot of nuclei found therein, because they're constantly churning out new proteins to replace old proteins and just basically keeping up muscle function. Besides the actin and myosin filaments to make up the sarcomere, you'll find other anchoring proteins which will anchor the sarcomere to the sarcolemma, known as dystrophin. You'll find some integral proteins called titin that actually holds the individual sarcomeres together. You'll also see mitochondria, of course, which shouldn't be a surprise, as you'll naturally need a lot of AT production for the muscle cell. Specialized myoglobin proteins that carry the oxygen just solely for the muscles, and of course glycogen for the actual energy stored to burn to produce more ATP. So this is probably the most important slide out of the entire chapter. So the basic functional component of a muscle cell is what's known as sarcomere, and we find these in all three types of muscle tissue. You find these in cardiac, smooth, and also skeletal, although in slightly different arrangements depending on which kind of tissue that you're looking at. So basically one sarcomere runs from one Z disc to the other Z disc. And basically what winds up happening when the muscle contracts, the two Z discs will actually come closer to what's called the M line. The M line is the center point of each individual sarcomere. And what you'll see is for every thick filament, you'll have roughly 6 thin filaments that are going to go around the periphery of the thick filament. So the thick filaments will be the myosin. Your thin filaments will be known as the actin. You'll have titin filaments that actually will anchor the thick myosin filaments to the actual Z disc. So these basically help knit everything together. We'll talk about what happens when those are destroyed, what kind of difficulties the muscle has to try and contract. So with those particular structures being set out, we also look at different bands that we usually see. And this is what causes the striations that we see, especially in skeletal muscle. So basically, if you look at the bottom portion of the slide here, you see what's called an A band. The A band is going to run down the entire length of the thick filaments. You're going to have the I bands, which are going to run basically where the area is only going to be laced with the thin filaments. You'll see no portion of the thick filaments whatsoever. You have the H zone, which is typically the area where you only see thick filaments. You'll see the zone of overlap where you see very simply thickened filaments run on top of one another. So each of these zones and the bands will actually change length, in particular when we see any kind of lengthening or we see any kind of shortening of the sarcomere. Okay, and there is a electron micrograph, a transmission electron micrograph of the sarcomere. So you can see this basically what it looks like in real life. So there you see the Z disc on either side, both the left and right of the M line. When the sarcomere contracts, you'll see the Z disc will move closer to the midline, and you'll see different areas will start to shrink. Typically, your A band, where the myosin filaments are, generally stays the same. The I band will shorten as the sliding filament mechanism of the actin filaments will slide on the myosin as they come closer together to provide the contraction. So different types of proteins that we see. So there are two main contractile proteins. You have actin and myosin. So basically the myosin will actually pull the actin filaments towards the M line. You've got two types of regulatory proteins. So these are the proteins that are responsible for binding with calcium. Troponin is the protein that binds with calcium that will form a change basically in how it's structured, and that will pull on the tropomyosin. What that will do very simply is open up a myosin bonding site to allow that to pair, and then basically will cause the myosin head to pull the actin closer to the M line. You have different types of structural proteins. So you have titin, which is going to anchor the myosin filaments to the Z discs. You go down to the dystrophin, and that is the proteins that are anchor the entire length of the muscle fiber to the sarcolemma. So again, your two contractile filaments will include myosin and actin. So your myosin is the thick filament, and these are typically shown with a tail and two myosin heads. It looks very similar to a golf club. This is where you're going to have what's called the cross bridge. That's what's going to bind with the myosin binding site on the actin. So the myosin heads will move, but the actual filaments do not. They will actually pull on the actin filament itself once the binding site is opened. Once the calcium will bind with troponin and will tug on the tropomyosin and basically slide out of the way to allow the myosin to bind. So here are your two regulatory proteins. As I mentioned before, you have tropomyosin and troponin. Tropomyosin is actually going to be the protein that's going to cover the myosin binding site when the skeletal muscle relaxes. Once there is sufficient calcium to bind with troponin, it changes shape and it will actually pull tropomyosin out of the way, as we mentioned before, and that will allow the myosin to bind. Structured proteins, the two most important ones here, again, are going to be titin. It's basically going to anchor the myosin to the Z discs. And you stick down to the dystrophin. These are the thin filaments that are integral to the protein, the sarcolemma. The other three, alpha-actinin, myosin, and nebulin, they basically will help, will bind first Z disc protein actin to titin. So it's a secondary protein to titin there. Myosin with the M-line protein binds titin to myosin. And nebulin, of course, will anchor the thin filaments to the Z disc. But again, titin and dystrophin are two most important proteins on this particular slide. So the sliding filament mechanism. So basically, the myosin will pull on actin, causing the thin filament to slide inward towards the M-line. So the Z disc will move as the sarcomere will shorten. So thanks to the structured proteins, there's transmission of force throughout the entire muscle, resulting in whole muscle shortening. So that means it's not a whole contraction. And that's very important to remember as we talk about muscle control. So the first question for you, very simply, so part versus whole contraction of muscle. So I want you to think about this. Is it beneficial or detrimental to have the entire muscle shorten but not contract completely? So what does that mean? So do we want all of the sarcomeres, all the muscle fibers to contract all at once? Or do you want only a part of the entire muscle to contract all at once? So here's what I want you to think about. You remember a few weeks back we had done the Play-Doh lab, and we talked about doing some fine-tuning, some fine motor control as we rolled out different structures, so on and so forth. So think of it this way. If you had the entire muscle contract all at once, and you try to do some fine motor control, you basically would squish that Play-Doh like you wouldn't believe. Another good example would be if you're trying to crack an egg, you want to do a very gentle type of crack, otherwise that egg is going to go all over the place. So there are some different types of diseases or some types of injuries that will cause a whole contraction of muscle, but so we don't use too much of the muscle all at once and use too much strength. We try and use a part of the contraction of the muscle for two reasons. One, for motor control, and then also for fine tuning, and also to make sure the muscle doesn't fatigue too quickly and burn through all its ATP stores. So to answer the question, number one is very important for fine motor control. So if you're holding something delicate, whether it be an egg, Play-Doh, or holding a baby, you want to actually use just enough force so you don't damage whatever you're holding. So you're applying only enough force that is required. Now, the flip side is, now if you ever have heard anybody that's ever been struck by lightning, or anytime they've stuck their finger in an electrical socket, basically all of that electrical energy can actually cause the entire muscle to contract all at once. As I mentioned before, the muscle and the tendon are actually stronger than the actual tendon attached to the bone, so you can actually avulse the tendon from the bone. So actually what happens is the periosteum and the bone itself will actually chip away and basically pull itself off the bone. So you can also cause a fracture depending if there's enough electricity going through the muscle itself. So that's one of the main reasons why you want to see a partial muscle contraction, and very rarely you want to see a full muscle contraction. The other reason why you want to make sure you have only a partial muscle contraction is you don't blow through all of your ATP store too quickly, and muscle can go into full fatigue. Now, occasionally you do see this though, where you see or you hear about people having unimaginable feats of strength, trying to move something heavy off of a level one or something like that during an accident. A lot of times it causes a lot of damage thereafter once the adrenaline starts to dissipate. So the sliding filament mechanism. So we're going to look at three different stages here. First, you see at letter A, basically a relaxed muscle. So you see there's nice even space between the eye bands, and also the H zone stays relatively around the same space on either side of the M line. You have a partially contracted muscle. You basically see the I band as well as the H zone starts to contract a little bit, but there's still some space still left over to contract. Letter C on the bottom there, you have what's called a maximally contracted muscle, where very simply it's almost as if the myocins are abutting up against the Z discs themselves. So there's minimal spacing. There's a minimal H zone there. There's also a minimal I band there. So respectively, there's a very small amount at the H zone where only the myosin filament heads are there, and also a very small zone, the I band, where there's only thin filaments there. So you see a lot of overlapping actin-myosin filaments all in those particular areas. Okay, so let's talk about the contraction cycle. So very simply, you're going to have ATP is going to bind with the myosin and it'll hydrolyze into ADP and a phosphate. So what that'll do very simply is, in conjunction with having calcium bind with troponin, what will happen is the troponin will pull away on the tropomyosin, exposing the binding site on the actin filament. Once the myosin head will dispose of the phosphate, the myosin head will bind to actin and form what's called a cross bridge. So there's been no conformational change yet to the cross bridge of the myosin head as of yet. We get to stage 3, it'll basically eject the diphosphate, and then that'll cause the power stroke, where the myosin cross bridge will rotate towards the center of the sarcomere, towards the actual end line, and push, or actually I should say pull, the actin filament towards the center of the sarcomere. Now that will stay there until a new ATP will come in and actually bind again with the myosin head. And once that ATP will bind with the myosin head, it will eject or release itself from the actin filament. So as long as there's enough calcium and enough ATP, this contraction cycle will continue. And of course, the calcium being released into the sarcoplasm is going to be dependent on whether there's a nervous stimulation or action potential traveling from the neuron to the muscle. So now I'll let you go through the contraction of the skeletal muscle cells on your own. So it's just a different animation, a different take on what we just talked about there, but you see everything match up there as well. But you actually will get to see the actual moving parts physically move through the animation. So welcome back from looking at the animation. So the first question I have for you here is rigor mortis. So actually the question really shouldn't be how does rigor mortis occur. The first question should be, when does rigor mortis occur? So very simply, rigor mortis, of course, happens when you're dead. So there's a couple key things that happen during this particular happenstance. So what winds up happening is a rigor mortis. What happens is roughly within 4 hours of death, up till about 24 to 40 hours worth, the body goes rigid. So in other words, the muscles will get locked in a certain portion of the actual contractile cycle. So if you think of it this way, there's one thing in particular that's missing during rigor mortis when you're dead. There's one particular part of the cycle where you get locked in. So take a guess. Could be 1, 2, 3, or 4. So is it where the ATP basically hydrolyzes to ADP and a phosphate? Is it where the phosphate will be ejected and basically allow the power stroke to occur? Or is it because there's no more ATP to bind to the actual myosin head to allow the release of the actin filament? So that's basically what winds up happening, is because there is no cellular respiration going on anymore. What winds up happening is you burn through the different stores like creatine phosphate. You start blowing through all the oxygen, so on and so forth. Once you blow through all the remaining ATP, there's no more ATP left to help release the bind of the myosin onto the actin filament. Now, given enough time after 48 hours, unfortunately, you start getting decomposition of the actual proteins themselves, and that's where the rigor mortis actually starts to relax. So again, it's because of the lack of new ATP that basically will lock the muscles up in stage 4 of the contractile cycle. So as you see here, once rigor mortis occurs, you see the sarcoplasmic reticulum will leak the calcium. The calcium will bind with the troponin. The myosin will bind to actin, and ATP hydrolyzes. So again, as I mentioned before in the previous slide, basically you do not have any more ATP to release the myosin from the actin filament. So you're literally locked in what's called the stiffness of death, or the rigor mortis. So let's talk about how the action potential traveling down the sarcolemma of the muscle couples with how the muscle either stays relaxed or it contracts. So basically, under a relaxed muscle, there is no action potential traveling down the sarcolemma. The terminal cisterns are closed, so no calcium is released in the sarcoplasm. So you see that contraption on the left. Now, on the right-hand side, as you see the muscle action potential travel down the sarcolemma, you see the terminal cisterns will open up and the calcium will flood itself into the sarcoplasm. That's going to bind to the troponin, it's going to pull in the tropomyosin and expose the binding site for myosin to bind to actin. So again, as long as there is a muscle action potential, the gate channels will remain open for the calcium, which will allow the sliding filament contraction to occur. And of course, as long as there is enough ATP in that area, it will stay contracted. So let's consider this. So what do you suppose would happen if you had too little or too much calcium? We'll answer too little first. So if we had too little calcium, let's say if the sarcoplasmic reticulum or the muscle cell itself did not have enough calcium, what do you expect would happen? And the flip side too, what happens if you have an overabundance of calcium? Let's say calcium is flooding into the muscles all the time. What would you expect to see, both from a micro and from a macro standpoint? Would you expect to see the muscle filaments to stay contracted? Would they contract at all? Or would you notice that the muscles themselves would be more flaccid, meaning they're very, very weak, or they would be very spastic, meaning very, very tight? And most of you have felt tight or spastic muscles before, so they're very rigid, they're very tough to relax as well. So let's examine first when we have too little calcium or calcium deficiency, otherwise known as hypocalcemia or a lack of calcium in the blood. If you have lack of calcium in the blood, there's a lack of calcium that gets into the actual muscles themselves. Usually it means you're not taking in enough calcium. You also have certain types of conditions like hypoparathyroidism, where basically the body does not pull enough calcium into the neighboring cells, into interstitial fluid. You have malabsorption, where your body simply cannot absorb the right kinds of calcium. Usually we see both calcium carbonate and calcium phosphate as two main types of calcium that we usually ingest. If you can't break those down, you can't bring in the right amount of calcium ions for proper muscle function. Different types of medication can also factor into this as well. So what happens to the muscle itself? Well, it causes cramps and it causes weakness. So if you look at it this way, if we cannot get enough muscle fibers to contract to do a certain type of work, whether it be lifting an object or whether it be trying to pull yourself out of seated position, you're basically going to have a minimal amount of muscle fibers available for the muscle contraction, and they're going to tear, they're going to get weak, you're going to strain those particular muscle cells, you're going to damage the filaments found within. So let's look at the flip side of hypercalcemia, or too much calcium found in the blood and also in the muscles. Now, you would think at first it would mean hyperspasticity, mean the muscle would be very rigid, and we actually find that not to be the case. It can cause spasms and pain, but it actually causes muscle weakness, because what winds up happening is the calcium, being a positive ion, will flood into the area and basically will block the flow or the flux of sodium ions crossing across the neural membranes. So therefore you cannot generate the action potentials that you normally could. So again, you run into actually the same kind of problems. The muscles get very strained over time, they get very weak, and it's very painful. So you see this in just the opposite kind of problems in hyperparathyroidism as well. Certain types of medications, which unfortunately I can't remember at this time, but you also see this with overuse of supplements. So people that take in too much calcium, usually in powder form, where the body takes in too much, basically cause this type of problem. Now let's talk about the length-tension relationship. So basically the force of the muscle contraction depends on the length of the sarcomeres just before contraction. We have a saying in the healthcare system, a long muscle is a strong muscle, a short muscle is a weak muscle. So basically if you look at the chart here, and there's going to be a nice animation that you'll look at in a couple slides later on, which does a very nice representation of this. If you look at the shaded area, it's just a little less than what the optimal length of the sarcomere length should be in micrometers. So basically you want to have just enough overlap between the actin and myosin filaments and still have enough areas in both the H zone and also the I band to allow the actual filaments to slide upon one another. So if you look at the shortened sarcomere just left of that optimal length where it's understretched, what happens is there's not much more room to allow contraction. So therefore the muscle cannot contract that much further. So there's certain structures in place that try and maintain the optimal length, which includes the tinin and dystrophin. That's why I mentioned those two structural proteins are more important than the other three that we mentioned on that one particular slide. They help maintain the proper distance between the actin and myosin filaments, as well as distance between the edges of the myosin and the Z discs. Now look at the flip side where things are overstretched. Now the problem is you don't want the muscle to stretch too much, where all of a sudden now the myosin completely misses the actin filaments, where they can't line up properly to allow the proper stroke and allow the sliding filament mechanism to occur. So you see this a lot of times in injuries. You see this a lot in a lot of slip and falls. You see this in a lot of different types of sports injuries, car accidents, construction injuries, you name it, where a lot of times these will also involve oftentimes some fractures of bones too, as you guys remember from ranges of motion. When something is hypermobile and goes past the normal range of motion, different types of structures start to break down. In this case, very cleanly, you can see the actin and myosin filaments. Once they go past a certain distance, there's no way they can get themselves together from that particular filament. Now, all is not lost, though. There is something the muscle can do, which I'll talk a little later on when we get into repair. Okay, the neuromuscular junction, the NMJ. So basically, this allows the action potential that travels from the axon of a neuron to basically pass through a synaptic cleft and allow a muscle contraction or an action potential to progress across the sarcolemma of the individual muscle fiber. So these are voltage-gated calcium channels that allow calcium to rush into the cell of the nerve synapse, basically. So what happens is you have exocytosis of acetylcholine, which is the main neurotransmitter. There's another one we'll get into, the autonomic nerve system, but for now, for skeletal muscle, acetylcholine is the main neurotransmitter that basically becomes secreted through exocytosis, which you should remember from a couple of chapters back, into the synaptic cleft. So this neurotransmitter is going to bind with ligand-gated sodium channels on the motor end plate. That's basically where the muscle begins and the actual axon ends, which will cause an influx of sodium into the muscle. That causes depolarization, and that allows the calcium release from the sarcoplasmic reticulum from the terminal cisterns. And then this whole process gets reversed again as the neurotransmitter gets broken down by acetylcholinesterase. So without this particular neuromuscular junction, you cannot have muscle contraction. So you see this with skeletal muscle in particular. There is some slight differences when we talk about cardiac muscle, because there's gait control channels that we'll talk about, and smooth muscle involves different neurotransmitters. But for the most part, for skeletal muscle, we're going to focus on mostly acetylcholine is the main neurotransmitter. So here we see a diagram very simply. You see an axon in the upper left-hand corner here. You see the axon will split into what's called a synaptic end bulb, which will synapse or cause a little spacing with the motor end plate. So the synaptic end bulb, excuse me, you can tell it's been a long day already. You can tell that the axon terminal at the synaptic end bulb will synapse with the motor end plate of the myofibril. If you look to the upper right-hand corner, see an enlarged view of the synaptic cleft. So you can see the voltage-gated channels for the calcium. So calcium rushes in, and then as you pass into the middle lower portion of the diagram there, you see acetylcholine. Once the calcium is fluxed into the axon terminal, basically acetylcholine is going to be released through the synaptic vesicle through exocytosis. It's going to traverse across the synaptic cleft and bind with the acetylcholine receptor. That's going to allow sodium to rush into the muscle cell. That creates the muscle action potential, and very simple, that will travel down the entire length of the sarcolemma to allow the calcium within the cell to be released from the sarcoplasmic reticulum and start the whole contractile cycle. And of course, as you see in stage 4, the acetylcholine is broken down to be reused again by the axon should another action potential travel down again through the axon. So this is a scanning electron micrograph of the actual neuromuscular junction. So each individual muscle fiber will have some type of synaptic cleft or some kind of connection with the axon. We'll see certain larger muscles will actually have one particular nerve that will synapse with, say, 3,000 different muscle fibers, whereas some muscle groups that require more fine motor control, in particular for say the fingers or the toes or even your vocal cords, you'll have one neuron for every 3 to 5 muscle fibers. So you have a lot more control, a lot more fine-tuning with those particular synapses at the neuromuscular junction. In fact, you actually even see some of the striations too, noticed on the skeletal muscle fibers there too, the overlapping actin and myosin filaments. So if we pair both sequences together, so if we have at number 1 here, we have a nerve impulse or we have an action potential traveling down the axon, the calcium will rush in. It'll cause the acetylcholine to be released through vesicles as it traverses through the synaptic cleft. It'll bind with the ligand-gated receptors, and that will trigger an action potential along the sarcolemma. That's going to basically allow the muscle to release the calcium found within the sarcoplasmic reticulum, in particular the transverse tubules. So that will allow the calcium within the sarcolemma, or the sarcoplasm, excuse me, to bind with the troponin. So that will allow the troponin tropomyosin set up to pull itself away from the actin filament. So as long as there is enough ATP, that will bind with myosin and allow the myosin to also bind with the actin filament. Once the ATP hydrolyzes and allows the power stroke, then basically it will release the calcium, it will release the ATP again, or actually release the ADP to allow ATP to bind again. Then constantly as this is going on, you have calcium pumps are constantly trying to go against the concentration gradient to draw the calcium back into the terminal cisterns. And once there is more sufficient calcium back in the cisterns and out of the sarcoplasm, you'll see the troponin and tropomyosin will start to slide back to its original configuration. And at that point, you'll see the muscle actually relax. So I'll have you look at this again. So basically, this is a nice animation of what I just talked about here, probably in more simplistic form, and probably a little more clearly, as most of you probably are laughing about right now. So come to the next slide once you're finished looking at this animation. So let's look at muscle metabolism, in particular how ATP is necessary to power the contraction cycle. This is where most of us, if you tend to specialize in, say, nutrition or even personal training, or if you do go into some of the other health fields such as nursing or to become a physician, this becomes very important. This is something I deal with day in, day out, especially with athletes. I deal with some people that play rugby. I have some power lifters. I also have a lot of endurance athletes. I have people that train for different shows as well. This becomes very important, especially with dealing with a particular type of exercise. But your 3 main stores are going to be creatine phosphate, which you're going to find in the liver, the kidneys, and the pancreas. You can take creatine phosphate as a supplement as well. There's different types. Probably the most well-known is creatine phosphate monohydrate, which comes in a powder form. In some individuals, this can cause some problems if you're taking too much. With just like any other supplements, taking too much can definitely disrupt things. But in particular, it can cause shutdown of the kidneys, especially the bone capsules. But again, I would definitely talk to someone a little more thoroughly about that, especially a nutritionist, to see if something like that is right for you, or if you're even doing an activity that requires you to bring in a lot more creatine. But that store burns out very, very quickly once we start using any kind of muscle contraction. I believe you have about 15 seconds worth of creatine phosphate already stored in the muscles locally. Then once you burn through that, you have anaerobic glycolysis, where it takes pyruvic acid to ATP and then converts the pyruvic back into lactic acid. This generally have a few minutes worth of anaerobic glycolysis, and then for any kind of long-standing or any kind of distance style of exercise, you fall into aerobic respiration. We fall into glycolysis, the Krebs cycle, and electron transport chain. Usually with aerobic respiration, though, the body starts burning through anything it can to keep muscle functioning as best possible. So again, the 3 types of production of ATP in the skeletal muscle. First, letter A, you see ATP being created from creatine phosphate. So again, you burn through this very quickly, around 15 seconds. If you take any kind of supplementation, you may be boosting up to a full minute, but there's not much. Once you get past about 15 seconds, that's when you start relying upon glucose through anaerobic respiration or anaerobic glycolysis. But of course, this builds up a lot of lactic acid, which can actually retard any kind of long-term muscle contraction. So you still generate some ATP, although to really kind of get a really high energy store, you want to transfer into the aerobic respiration, which can last for minutes to hours. So with this, it draws oxygen from hemoglobin, also myoglobin. That's going to be the specialized carrier protein found in muscle fibers. You're going to take pyruvic acid from glycolysis. You're going to basically break down your fatty acids from your adipose cells, but also during that time too, especially with long-term exercise, you're also breaking down amino acids from proteins. So basically, you're kind of eating yourself up a little bit, almost literally, for long-standing exercise. This is why it's so important for a lot of us if you're going to do any kind of long distance or any kind of long-standing exercise. You want to supplement with enough protein, enough carbohydrates, so you're not breaking down the same kind of muscle you're trying to use during the activity. Okay, so again, a little more in-depth through the animation here for muscle metabolism. Go ahead and look through that at your own leisure, and I'll see you at the next slide. So muscle fatigue. So basically what this means is you cannot maintain the force contraction after prolonged activity. It could also be after a very short strenuous exercise as well. So this is due to a couple factors. Number one is not enough release of the calcium from the sarcoplasmic reticulum. There could be depletion of creatine phosphate and or oxygen, other nutrients. So you can never really get to the aerobic respiration. You can have buildup of lactic acid and ADP. So you don't have the ATP to hydrolyze to release that phosphate and release that energy. Or sometimes there's simply not enough release of acetylcholine at the neuromuscular junction. So let's talk about oxygen debt. So why do you continue to breathe heavily after a period of time after stopping exercise? So this is basically what we refer to as oxygen debt or recovery oxygen uptake. So basically your body fights to basically restore itself or restore the energy stores that it has. So it replenishes creatine phosphate. It takes time to convert your lactic acid back into pyruvic acid. So again, it can be used again for your heart, liver, skeletal muscle. It's also known as what's called the afterburn. Also winds up happening is you're trying to reestablish levels of oxygen to bind with myoglobin. Your glycogen, however, that's something that usually comes in from your dietary carbs. That's why it's important to eat something, usually within, depending on what kind of articles that you read, anywhere from a half hour to even a couple hours after your workout. So again, your body isn't cutting through your protein stores more than anything else. You want to use the proteins to actually build up the muscle. You want to use carbohydrate restore to basically build up your glycogen again. So let's talk about control of muscle tension. So basically a motor unit is very simply a motor neuron from its axon as it synapses or innervates certain muscle fibers. Now, as I mentioned before, some of the larger muscles or the muscles that do a very specific thing, in this case the gastrocnemius, and those of you who don't know what that is, that's one of the major calf muscles. So this is very important for locomotion in particular. So you may have one motor neuron that actually will synapse with 3,000 individual muscle fibers. You don't need a whole lot of muscle control for the gastrocnemius because it's more of a power muscle. However, for more fine tuning, such as the laryngeal muscles, when you provide speech patterns, you basically may have one motor neuron only controlling 2-3 muscle fibers at a time, which, as you can see, can give you a very, very, very fine-tuned motor control here. The average is about 150 per neuron, but it does vary quite a bit depending on the muscle group that we're talking about. So when we talk about the strength of muscle contraction, however, it basically depends on how many motor units are actually being activated at once. Now, twist contractions are going to comprise different stages, different periods, where all the muscle fibers in the motor unit will respond to one action potential. So basically, it starts off with a latent period. So that's the actual time it takes for the actual action potential to travel to the axon, have the acetylcholine span the synaptic cleft, and actually allow the calcium to actually be released, and then actually bind with the troponin. This can be known as the latent period. So It's just the amount of time that it takes to traverse those small distances. Once the calcium will react with the troponin, that'll start the contraction period. And as that's happening, as long as there's not another action potential being generated, you have the calcium being pumped back into the terminal cisterns, and you have the ATP being replenished at the same time. The muscle relaxes. Now, very similar to when you have a nerve fiber action potential, there's a refractory period. There's a time where there absolutely cannot be another muscle contraction during that refractory period. Now, for most of us, it's barely perceivable, but it is there. So we're going to talk about the different types of stimulation here. One is dealing with the frequency of stimulation. So a wave summation occurs when you have a second action potential will trigger muscle contraction before the first contraction has finished. So usually this means that one builds upon the next, gives you a stronger contraction. So we have two types of what we call tetanus. One's called unfused and one is called fused. So a fused tetanus is when muscle stays contracted, there's no relaxation. Each individual muscle fiber may not contract all at once. You may have muscles that are fire off left, right, front, back, side to side in intervals, but the entire muscle will stay contracted. An unfused tetanus is when you see a muscle wavering or almost like a twitch, so to speak. A lot of times you'll see this when people have certain types of muscle spasm or a little twitch in the eye, twitch in the finger. That's what's known as unfused tetanus. So very simply, the muscle relaxes for just a very short time before it contracts again. So visibly, or at least measured on certain types of myographs, you'll notice that the muscle does have relaxation period. So this is a picture of a myogram. I have something very similar to this in my office. It's not very precise. We use it more so for patient education. But if you start off to the left here as single twitch, you notice there's an action potential generated in blue that's coming off of the axon. So you see the action potential or the force contraction just above that. So it shows an increase in the force contraction. Then it relaxes, and then letter B, you see two sequential action potentials where right before the first action potential or the first force contraction stops, it builds upon that and causes a stronger contraction or higher amplitude in the force contraction. Then you see the letter C, the unfused tetanus. So you see your periodic action potential, where each one you see a little bit of wavering back and forth. The muscle does relax for a split second, contracts, relaxes, contracts slightly. So you see almost like a twitch going on in the muscle. And last is seen in letter D, that's called a fused tetanus. So you have a rapid response of action potentials one right after the other, where it does not allow the muscle to relax. You see a sustained contraction, as you see the force contraction on the red line there, where it hits a maximum point, levels off, and then finally the muscle finally does relax. Now, motor unit recruitment. So basically that will control the number of active motor units in a certain muscle group. So what happens is, this is where the fine tuning comes into play too. When we start a particular motion, let's say if we're lifting something, if you ever lifted something off the floor, you tend to use your weakest motor units first, and then you start to slowly build up and actually add more motor units to help build a stronger contraction. So a lot of times what happens is if you're trying to hold something in a very long period of time, you'll get different motor units will contract alternately, one right after the other. So the entire muscle will stay contracted, but individual muscle fibers will not contract very long. One will shut down, one will relax, another will contract. Those of you who are in ROTC, when you stay at attention, this is the phenomenon that keeps you upright without allowing you to go fatigue and fall over. Without this, the muscle fibers will fatigue very quickly, and it'll be very tough to hold any particular static position. So the better explanation for this will be available in the animation here. Take a look at that, and then meet me on the next slide. All right, so muscle tone. Muscle tone is very important. This is something that I deal with in my office quite a bit. So muscle tone should have a certain amount of tension within the muscle. When you have too little, it's known as flaccid. If it's too strong, it'd be in a spastic position. A lot of times the spastic conditions can be very painful. So when I see a muscle that is too flaccid, meaning it's too weak, it means you're not getting proper contraction of motor units. So even when you're asleep or you're relaxed, there should be a little bit of muscle tone left in a particular muscle. Naturally, if you're doing any kind of activity or lifting weights, you'll see more motor activity, and the muscle actually will harden as it goes in contraction mode. But there should be a certain level of muscle tone on each muscle. So a lot of times what we see is if a particular nerve that innervates that muscle is either compressed, and it doesn't take much, it only takes about the weight of a dime to disrupt an action potential down the nerve, or if the nerve is overstretched for certain types of injuries, or even cut, God forbid, then that muscle loses the innervation, it loses its tone, and therefore becomes flaccid. So let's talk about different types of contractions. And there's a third one here that I'll talk about as well, but you won't be tested on that. So we have isotonic versus isometric contractions. So isotonic, most of you should be familiar with, is what we normally see in the gym. It basically means that tension is constant while the muscle length changes. So in other words, most people know what a bicep curl is. So if I took a 10-pound weight and I held it relaxed with my arm straight down, and I bent the elbow or flexed my elbow, that is what's known as a concentric tension, or a positive or a shortening of the muscle, but the weight stays the same. In other words, the tension or weight stays the same at 10 pounds while the muscle length changes, so it shortens. The flip side, doing the negative, is very simply, if I held that same 10-pound weight in the flexed position in the bicep and I very slowly let it relax, that's called a negative or eccentric contraction. For a lot of people looking to build muscle, this actually works very well. Because if you remember when I talked about muscle recruitment, use more muscle motor units during the concentric or the positive shortening versus the eccentric or negative. This usually will, if you remember from Wolff's Law, for bone, there's a very similar law used for the soft tissues. So you're basically alerting the body to basically lay down more muscle tissue when you do the eccentric or negative contraction. Now, isometric is a little bit different. Those of you who have used exercise bands. So if you imagine this, the exercise bands are basically huge rubber bands. So with this one, if you try and hold a muscle in place, so if you think of it this way, those who have ever gone water skiing before, you're trying to hold yourself upright and holding onto the cable with both hands. So imagine if someone's holding on the cable and it's elastic, and they are pulling harder on the band. The band actually will increase the strength to maintain your position. So in this case, the muscle contracts but does not change in length, but the load does become stronger. So in other words, again, the muscle, you're basically holding a static position, and they're increasing the tension on the band, which is called isometric contraction. Now, the third one is called isokinetic, and you won't be testing this is more so for your information. Isokinetic is a combination of both isotonic and isometric. This basically is you using the exercise band and you actually contracting the muscle. So in this case, if you're doing a row motion with an exercise band that might be attached to a wall, you're going to be contracting the muscle. And as it contracts, that band is going to increase the tension. So it's kind of like the best of both worlds here. You're actually tricking the muscle in both ways. You're shortening the muscle and also increasing the load at the same time. This is usually the fastest way to build any kind of muscle strength. Here's a nice little shot of this particular person showing isotonic versus isometric contractions. So A and B shows both concentric and then eccentric contractions, but they're both isotonic. So concentric would be actually moving in a flex position, and then eccentric would be going negative or lowering the book into relaxing from the flex position. This is not the same as extension because you're slowing the process down, so it's eccentric. If you're pulling a weight forcibly under tension, then there'll be a concentric contraction on the tricep, but this only shows the bicep. Letter C is an isometric contraction, so if you notice he's holding the book out steadily there. In this case, he's using mostly his anterior deltoid and part of his bicep and also some wrist flexors. So in this case, the book does not change in weight specifically, although most of you, if you notice, if you try and hold a book out that long, the muscle does get fatigued over time. You can't hold that book up forever. So let's talk about red and white muscle fibers. So this is where we get into your dark meat and white meat that so many people are familiar with, especially when we go for Thanksgiving dinner. So we also have dark meat and white meat all interspersed throughout our body. Depending on what we're asking the muscle to do, the dark meat are usually going to be the more higher sustaining positional muscles or any kind of distance or endurance muscles, whereas the white meat typically are going to be your quick motion type of muscles. So we see different characteristics of each. So the red muscle fibers are typically going to be very high in myoglobin, have a lot of mitochondria, and have a lot of blood vessels to help maintain or sustain endurance type of activity. Whereas the white muscle fibers are going to be lighter because there's going to be lower levels of myoglobin, of course lower levels of mitochondria, and lower levels of blood vessels. So these will tend to fatigue a little more quickly. Now we have one that's going to actually be in between these two types of the red and white muscle fibers. We have more descriptive terms for these though. We have what's called slow oxidative, which are going to be your red meat fibers. These are going to be your types of fibers that are going to be, again, for endurance. You can have fast oxidative glycolytic. These are going to be your in-between muscles. These are going to be your actual lifting muscles to move things back and forth. Your fast glycolytic, these would be the ones that are the most white fibers. These are for your quick burst motions. So these are kind of things like you're trying to catch a ball, or you're trying to push stuff out of the way of a speeding car. These are very quick moving muscles and generally will fatigue relatively quickly. So basically the ratio of what kind of muscle fibers that you have is all based on genetics. So unfortunately what you're born with is what you're born with. So you may have more slow oxidative muscle fibers than say the person next to you, or you may have less. This is not something you can change. Unfortunately you're born with what you got. So very simply, there's slow oxidative muscles. These are your long distance running muscles or your endurance muscles. You tend to be dark red, high myoglobin, slow speed of contraction, fatigue slowly. You're basically going to have, it's going to be the slowest out of all three of them for ATP generation aerobically. Your fast oxidative glycolytic would be medium levels of myoglobin. Like I said before, these are going to be your in-between fibers. Tend to be dark red. They have fast ATP generation aerobically. Moderate fatigue, fast speed of contraction. These are muscles for walking, sprinting, also for doing kind of weightlifting as well. Your fast glycolytic are going to have low myoglobin, be white in color. They'll contract strongly and quickly. These are your burst motions. So we think of strength training. This is going to be like your clean and press, your jerks, those quick motions in particular. But these muscles also tend to fatigue very quickly. This is where the creatine phosphate we had talked about before. If you are supplementing for it, this is probably the main muscle group that you'll be using them more so for. Now, exercise and skeletal muscle tissue, just as we mentioned before with Wolff's Law. Basically, there's another law, which I believe is called Davis's Law. So Davis's Law is the Wolff's Law, except applied to soft tissue. So this is the whole reason why we always stress that it's important to work out. And workout can basically mean anything from cardiovascular exercise, which actually improves, naturally how well your heart beats, and also the efficiency of the heart. Doing strength training as opposed to doing cardiovascular is also very good for building either strength and/or endurance in certain muscle groups as well. So you can actually transform the muscle groups as far as the muscle fibers. You can actually increase the production of actin-myosin, blood vessels, mitochondria, but you're not building more actual muscle cells. You're actually taking the muscle cells and making them bigger. We always talk about stretching. This is always a buzzword, especially in my line of work, because people always ask me, is it better to stretch either before or after? And it really comes down to the individual. For most patients that I see, stretching is used best after heating. Now, for some people, they may do a quick, like 5-10 minute cardiovascular exercise, get the blood flowing, then stretch thereafter. I will say this, though, is not a good idea to stretch cold or when you first get out of bed. You want to get warmed up a little bit first. Some people even go as far as doing the stretching after they're done the full workout. For example, as a runner myself, I generally like doing stretching when I'm done my run. So it's almost like a prep before I do my next run the following day or following week. But again, it all comes down to the individual. Now, strength training, we know the obvious benefits, increased bone density, as we had saw from the animation before, on the femur of the athlete versus the femur of, say, one who is osteoporotic. Naturally, increasing muscle mass will give you a better metabolism. It's more logs in the fire. So if you're looking to, for someone that's looking to, let's say, burn fat, you do need a certain amount of muscle strength to help kind of burn that fat a little more efficiently. But also there's other benefits too. It also obviously will decrease physiological stress. For most of us now, especially be stuck at home, Most of us are spending a lot more time in front of the computer. Some of us in less than ideal positions. For example, and you'll see when I do my videos for some of the labs coming up, I have a makeshift office down in my basement because of one of my kids is using my actual office in my house. So you're working on very inefficient workstations. So that also will tie in the physiological stress and also the psychological stress too, which I won't get into too much because that deals with neurotransmitters, so on and so forth. We will talk about that when we get into brain anatomy, but for now, let's just talk about the muscular benefits. So it does help decrease psychological stress. So if you haven't taken advantage of it yet, I definitely encourage you to get outside. If you have any kind of workout equipment at home during this stay-at-home period, I certainly encourage you to do so as much as possible. Okay, so that will wrap up most of what we have talked about for skeletal muscle. We'll talk about aging of skeletal muscle in a little bit, but let's talk about the slight differences with cardiac and also smooth muscle compared to the skeletal muscle we just talked about. So cardiac muscle, as we said before, has a very similar arrangement to skeletal, except it has intercalated discs. So there's going to be an extension of the sarcolemma, and it's going to be anchored by desmosomes. So these are your gap junctions. These are going to be contracted by electrical impulses. And you'll see, well, actually, we'll answer the next question in a quick sec here. So desmosomes are going to have intercalated discs, going to stitch each individual muscle fiber together. So you're going to have gap junctions, which will help transmit action potentials from cell to cell. This allows a prolonged contraction due to prolonged calcium and sarcoplasmic delivery. What's kind of intriguing is you have what's called autorhythmic muscle fibers. You have two nodes that can be found in the cardiac muscle, the SA and the AV node. If you were to take individual muscle fibers and put them in, I forget what kind of solution, I do apologize, they'll each beat to their own rhythm. Once you join the muscle fibers together, within a few seconds, they'll all start to actually rhythmically beat together all at once. So once they link up, they will actually function all as one. That's what the primary information, the primary reason around the SA and AV node is, which will have one portion of the cardiac muscle contract, the next one contract just a split second thereafter to allow proper flow from the atria to the ventricles, and then therefore out different parts of the body. You can get hypertrophy with cardiac muscle. You can get a physiological enlarged heart from an athlete, which really means it becomes more efficient. You're building more muscle fibers, but usually you see a lower heart rate with that. You can get hypertrophy in cardiac muscle too with increased loads. Now this could be due to obesity. This could be to COPD. So basically it's tougher to push the blood flow through certain areas. That's the kind of hypertrophy you don't want. That's the kind of thing that usually requires some kind of medical intervention at that point. Our smooth muscle. Now, smooth muscle is going to be a little bit more specialized because we want to see more sustained contractions. They're usually contracted more so by hormones and also by, in some cases, by different neurotransmitters. Can be acetylcholine, can also be some other ones as well. They can shorten the stretch to a greater extent than what cardiac and skeletal muscle can do. So with smooth muscle, we see if you have some single unit muscles with the neurons, you may see, like along certain capillaries, you might see multi-unit smooth muscle tissue around some of the larger areas, like say around the esophagus or around the larynx and so on and so forth. You'll see some very fine motor control here, but again, this is going to be under involuntary control. Now, what you do see with the sarcolemma and also with the sarcomeres in a relaxed and smooth muscle fiber, they're going to be interspersed and also connected by different intermediate filaments. So when these contract, they contract more similar to almost like an inchworm or a planaria, if you guys remember this from high school biology. So the entire muscle contracts, where basically like it becomes very thin in a relaxed position, and then actually will become very short and squat when it comes contracted. So naturally, when it contracts, it actually will usually vasoconstrict blood vessels or constrict different tube organs and so forth. But these are generally going to be for more of a sustained contraction. Now, as far as regeneration of muscle tissue goes, once skeletal muscle fibers, now we're talking about skeletal muscle fibers in particular, and to some extent cardiac too, Once it's fused, it does not undergo mitosis anymore. So what ends up happening is the amount of muscle cells that you're born with for the heart and for the skeletal is what you have. If you ever lose those picker cells, you're basically trying to call upon the other cells you have to basically hypertrophy or enlarge, create more muscle fibers within the cell. So increase the fibers, in this case hyperplasia, but you're not replacing the cells anymore. Which unfortunately for a lot of people, especially with if you're losing heart cells from necrosis or whatever else, from heart attacks, you get scar tissue in those areas. So a lot of times the muscle does not contract. Now, what's intriguing is smooth muscle, because it has neighboring parasites. These can actually undergo mitotic division and replenish smooth muscles. We get down to the capillary beds, then there are more stem cells that replace the individual smooth muscle cells. This one's easy. Developed muscle cells, they all arise from the mesoderm. That's the only thing embryologically I want you to know. They all come from muscle cells, from the mesoderm. All right, so it's no surprise as we age, we start to lose muscle tissue. And unfortunately, it's a very quick slide that starts around age 30 and continues to age 50. We lose about 10% of the muscle tissue. It starts being replaced by fibrous connective tissue, in this case, scar tissue. And in some aspects, especially if there's a lack of exercise, adipose tissue. By the time you're 50 to 80 years of age, you lose another 40% muscle tissue. This is why it's important for a lot of the population to maintain some level of activity as we age. If you can keep up a certain level of activity, you can staff off both of these drops in muscle tissue. But we do see that muscle strength and flexibility does decrease. Your reflexes all start to slow down. You also have to notice that the slow oxidative muscle fibers start to increase. So for some people, most of your endurance style of exercise, that's why usually we'll ask our elderly population to at least walk as much as possible and use some weights, but not the heavier weights, because your slow oxidative fibers, they generally will stick around for a long period of time. All right, then that's chapter 10. Be sure to answer those 3 questions that we talked about before. You'll pick up next on chapter 11 with the entire muscular system, and I'll give you some tips on how to navigate that particular lengthy chapter.